Hello everyone, I'm Sergio Mena, graduate student at the Hashemi Lab at Imperial College London, and in this short video tutorial I'm going to show you the Analysis Kit, which is a new web application for the visualization, filtering, and data analysis of fastest scan cycle voltammetry signals. Now, fastest scan cycle voltammetry, or more commonly known as FSCV, is a technique that allows us to monitor neurotransmitters in vivo and in vitro in a sub-second time scale. Now, if you intend to use this application, or if you know the Hashemi Lab, you likely know what FSCV is, but otherwise feel free to access a link in the comments below that will link you to a comprehensive review of what FSCV is. Now, this web application intends to offer post-analysis tools in an open source and freely accessible web application. The application has been tested with 3 major browsers: Google Chrome, Mozilla Firefox, and Microsoft Edge. And one of those and a PC is pretty much everything you need to get started. So let's begin. So the first thing you will see when you open the application is a homepage with a brief description of the web application and a few links to open the different analysis tools. Now, today we're going to focus on the analysis of FSCV data, which can be opened with the following button. Once you open it, a new window will emerge with the main application of FSCV Analysis. The application allows you to upload several FSCV signals simultaneously in text, TXT format, CSV, comma-separated files, or Excel spreadsheets. The structure of the data in the files is a 2D matrix with the current values of the color plot. Each column is a cyclic voltammogram of the color plot, while the horizontal lines, the rows, are current time traces at a particular potential. In the text files, samples are separated horizontally with tabs and vertically with new lines. Example of these files can be downloaded in the documentation section of the homepage of the application. Before including the files onto the application, there are 3 important parameters from your acquisitions to check. The first one is the acquisition frequency of your acquisition card. This can be read from the specifications of your acquisition card, or it can be also simply calculated from the number of samples in each of your cyclic voltammogram divided by the time it takes for the voltage program to run. The other 2 parameters are straightforward: the cycling frequency, or frequency at which the voltage waveform is applied, and the units of the current values in the matrix, which usually for dopamine and serotonin are nanoamps. After deciding which files to upload, wait for the green tick to show, which marks that all your files have been uploaded correctly, and then click Include. After that, the first color plot will show into the graph, and you can scroll between color plots with the Previous and Next buttons at the top of the application. The Embed button allows you to embed the current values and change between UPAC and USA convection. Background subtraction can be achieved in the application, and is done by selecting the sample CVs in the color plot that reflect a period of non-activity. Those CVs will be then averaged, and each CV in the color plot will be subtracted with an average, which represents the background current. The signal-to-noise ratio of the acquisition can also be calculated in a way that is similar to the background subtraction, by selecting a section of the color plot from when the noise is estimated, which should be a period of non-activity. The color plot can be shown in 3 different formats: heat map, which is what we usually know as color plots with false color, 3D graphs, and contour plots. The user can also select a wide range of color palettes, and in the graph configuration, the maximum and minimum values of the color palette can be changed. After background subtraction, the next thing to do is to filter your signals. This can be achieved by 2 different methods in the application: Gaussian convolution smoothing, or 2D Butterworth low-pass filtering. For convolution, which is the simpler, there are only 2 parameters that need tuning. One of them is the standard deviation of the Gaussian kernel. The larger the Gaussian kernel, the stronger the filtering. And then the number of repetitions, which is simply the number of times you apply the kernel to the color plot. 2D low-pass filtering requires a bit more thought. Here, there are three main parameters that can be changed: the order of the filter and the cutoff frequencies in each axis, in terms of the perfect percentage of the maximum frequency. The lower the cutoff frequencies, the stronger the filter. Additionally, the order of the filter will define the transition band of the filter. A high order will make a steeper transition, which is commonly a good thing, but we will also increase the likelihood of having filtering artifacts, such as the ringing artifact. The Fourier spectrum can also be seen in the application, and the configured filter can be superimposed into the Fourier spectrum to know which frequencies you will be attenuating. Once you have filtered the signal, you can apply changes to transmit these changes to the data stored by the application. Additionally, the filter color plot can be exported in text or Excel formats. Usually, the physiological information from signals is extracted from current time traces at voltages that correspond to the Faraday voltage of our molecule of interest. This can be interactively achieved in the application by toggling the graph selection button and clicking on the graph in the point at which you would like to extract the trace. This automatically graphs the selected horizontal and vertical lines which correspond to the current time traces and the cyclic voltammogram into the left graph. Each of the traces is assigned a unique color and the name of the file of origin. If several traces from the same file are added to the application, a number would also be added. When all your traces of interest have been extracted, you can apply a calibration factor to convert the current traces into concentration traces. The concentration units and a name for the trace are also required, as well as selecting the curve that you want to calibrate. When you do this, the application will automatically calculate the maximum amplitude of the release and the clearance rate based on a linearized exponential fit of the reuptake curve. The points used by the algorithm to fit the exponential curve can be changed manually, and additionally, the fit can be also improved applying a gradient descent algorithm to optimize the exponential parameters. This usually provides a much better estimate of the clearance rate of the neurotransmitter. The calibrations, as well as the average trace, can be extracted in an Excel file. In a detached application, the concentration traces can also be fitted with a Michaelis-Menten reuptake kinetics model. Two main parameters describe the transport kinetics of a neurotransmitter: Vmax, which is the maximum speed of the transporter, and Km, which is the concentration of the neurotransmitter at which half of the maximum speed is achieved. Two different models can be fitted in the application to the experimental signals: a one reuptake mechanism model, extensively used for dopamine, and a two reuptake mechanism model, commonly used for serotonin. Any of the calibrated transients, or the average of all of them, can be transferred to the kinetic reuptake window. We will start showing the one reuptake mechanism first with a dopamine transient. A first thing to notice when opening the application is that there is a panel to import signals. This allows you to import the average concentration trace that can be exported from the previous window, but there is no need to use it if you open the window from FSCV Analysis. Another thing to notice is that there are 6 inputs, but only 3 of them will be fed to the signal: Vmax, Km, and SP. SP stands for the constant neurotransmitter and release per stimulation during the experiment. The description of each of the parameters and what they mean can be found by pressing the information button. The two graphs show the model and experimental trace and the release term of the differential equation. The other 3 parameters are related to the stimulation paradigm following during the experiment: the start of the stimulation, the frequency of the stimulation, and the number of pulses. Once those are set, it is time to optimize the kinetic parameters. In the application, a range of accepted values can be provided, so that the optimization algorithm does not look for a solution that will not make physiological sense. Each of the parameters can be optimized individually or all of them at the same time. The number of iterations in the algorithm and the learning step also need to be provided. A higher number of iterations will take longer, but the final solution will be more likely reached. When the user is happy with the fitting, which can be assessed visually, and with the estimation of the room mean square error, the model can be exported into Excel. The application to fit the two reuptake mechanisms has a similar structure, but the fitting method is different. First, the release term is manually modeled and not dependent on experimental parameters. An additional autoreceptor term is present in the equation, which acts as a representation of the negative feedback control of the release produced by autoreceptors. These two are modeled by a series of time breaks and rate of change that can be visualized in the second window as you modify them. There are 4 kinetic parameters to optimize, representing the two reuptake terms. The parameters are set by default for the reuptake 2 to have a hypercapacity but low affinity. In the configuration panel, the weights of the two reuptake mechanisms can be changed and configured to change above and below a certain threshold of concentration. Commonly, the first reuptake takes place at high concentrations of serotonin, for which it will be set as a positive value above a concentration threshold. Additionally, an estimation of the basal concentration of neurotransmitter and basal release to match the basal concentration is required. For serotonin, this has been estimated to be around 50 nanomolar to 60 nanomolar for control animals. Finally, just as it is the case with the one reuptake application, the optimization window allows you to pick the parameters to optimize within a specific window and select the number of iterations and learning rate. Once the fitting is done, all the traces and parameters can be exported into an Excel spreadsheet. And that's it for this short video tutorial. I hope I have convinced you to use the application in your everyday analysis of FSCV signals. Just a couple of additional comments. If you're interested in looking at the source code that runs the application, it can be freely accessed in a repository that you will find in the comment section. Additionally, if you would like to give us suggestions on new features of the web application, report bugs of the web application, or any other questions or comments that you might have about the application, feel free to contact the Hashemi Lab on Twitter or me directly by email. And thank you very much for watching the video, and I hope you have a nice day.